All right, so this is your 3.2 part 2, the cell cycle. We talked about in the previous part mainly the mitosis and the way the microtubules do segregate the chromosome. And today, instead of that, we are going to talk about the real cell cycle instead of the mitotic division that we talked about, that the small part of the cell cycle, which was right here. We are going to talk about the interphase, which will be the G1, S, and the G2 phase. So the most important part is that once the cell is done with the mitosis, we talked about that, it enters into the G1, or it can go into the G0 phase, which is the quiescent stage. And we're not going to talk about that, but how we are going to see that through the G1, how it goes into the S and the G2. And in each phase, there is a checkpoint. And what we mean by the checkpoint, that as the cell proceeds through each of these stages, it asks a series of questions. And the way it asks the series of questions, when the cell goes through these phases, it asks a series of questions. And this ask a series of questions means that there is a certain ways that a particular protein or an enzyme behaves in particular cases, and that just leads to ask these question type, that what do you mean by that? So we're going to see that how this actually works and what are these questions. So let's take a look that when we are looking at this cell cycle, how it is controlled. So from the M phase, it exits from this phase, and we call this one as a metaphase checkpoint. It exits from the metaphase where the cell goes into the anaphase, telophase, cytokinesis. And then we are going into the G phase, the G1 phase, the gap 1 phase, and we are going to talk about that. In this phase, towards the end, which is kind of a late G1 phase, this usually is considered like there is no specific rule, but why it is chosen is just a consensus of the scientists, that we usually tell that this is the start point of the cell cycle, and if the cell cycle kind of stops right there. You just first cycle right here, and this ends right here. So this start is also called a G1 checkpoint, and here a series of questions is asked. And the questions is that, is the cell big enough? What we mean by that once it exits from the mitosis, the cell is just divided, and usually it's much smaller and doesn't have the required amount of organelles and everything, and the cells grows in the G1 phase. So before it goes into the next phase, at here in this checkpoint, it is asked that whether it's big enough, is the environment favorable? And what we mean by the environment, is there enough amount of nucleotide to synthesize the DNA? Is there good amount of an amino acid pool to synthesize new proteins? So all these biochemical constituents of the cells are checked. That is what we mean by the environment, favorable or not. And one of the most important part is that in this checkpoint is that once the cell is done with the mitosis, goes through the G1, where there is nothing going on with the DNA as such, other than this growing and making of the different proteins which are required in the next phase. Is there any DNA damage? That is an important thing that is being asked. If there is a DNA damage, the cell will not move into the next phase. It will halt right there, and we're going to talk that in detail. If everything is okay, it moves into the S phase, which is the synthesis phase, and here the DNA replicates. And then you will again have another checkpoint, which is a G2 checkpoint, where after the replication of the DNA, the questions are asked, is all DNA replicated? This is absolutely necessary for division into two daughter cells, which will equally get the equal number of chromosomes. And the DNA is needed to be replicated, not only completely, but also without any damage. All these things are going to be checked here. And also it's going to ask, is the environment favorable for the division or not? Is the cell big enough? Is the DNA damaged? Also, very interestingly, that is the DNA is the environment favorable, not only biochemical. We actually have seen that if you in the culture keep the cells in a colder temperature, it slows down the cycle. So it can sense the temperature even, that whether the temperature is favorable or not for the division as well. So all these questions are asked throughout the cell cycle. And then in the metaphase, the questions are asked, the checkpoint, are all chromosomes aligned in the spindle? So think about that, that we all know that from the previous class that in the metaphase plate, all these chromosomes are aligned. And that is very, very important. If they are not aligned, they will not be going towards the pole, and whether they are with one specific side of the spindle or the kinetochore microtubule, that is important, and also the environment favorable. So these are the reason that this is how the cell cycle is controlled. And this is a biochemical control. What we mean by that, these are a series of proteins as well as enzymes that control this system. So let's take a look at how this was discovered. Before going into that, let's take a look at a very basic question right here. So let's start that. Your first question is, refers to ordered series of events that lead to cell division and the production of two daughter cells, each containing chromosomes identical to the parent cell. This is mainly if you have not been in the last class, and I'm sure that everybody will be able to kind of understand the basic concept of that. "15 seconds. Is everybody doing all right with their internet speed? Is anyone having issues logging into Reef? So I see so much about, so far about 80, 81 responses. Okay. Great. All right, so I'm stopping about a 1.5 minutes now. So yeah, I mean, like if you just cannot just go in there, you can just always take a look at these questions. I'm going to talk about that. So I'm stopping that. And of course, I mean, like this is a very general question, and I just asked if you were not there, that the cell cycle is the answer, obvious. And that's about it. So let's move on. There is nothing to explain here. It's a very straightforward question, no trick, nothing. So now we were talking about this biochemical control and how it was discovered. So what the scientists did, and these experiments were done long, long before, they took a cell in the culture, which is a mitotic cell. So what you are seeing that the chromosome have already condensed in this stage. And also they have taken another cell, which could be from a G1 phase, S phase, or the G2 phase, where you can completely see the nucleus. There is no condensation of the chromosome. And they fuse these cells. There are several ways you can do that. You can just put them in a cuvette, pulse it with high voltage electricity for a short period of time. They will fuse. It's called electroporation. You can use a polyethylene glycol that fuses membranes as well, but somehow you fuse the cells. We have seen that, excuse me, we have seen that when we were trying to develop monoclonal antibodies, how you make these myeloblast cells fuse. So once you fuse these two cells, what is interesting that is happening, you have already some of these chromosomes which are condensed, but along with that, all these DNA that came in the chromatin format, which were just hydrated and elongated thread-like structure, they started to do the condensation. So what the scientists at that time mentioned or deduced from the hypothesis that there must be in this cell, the green cell, must have something that they called as a mitosis promoting factor, or MPF. So they did not know at that time what it is, but they pretty much argued that there must be something which is causing the condensation of the chromosome. And of course, they were discovered one at a time. There are many, many of these mitosis promoting factors. Not only that, throughout the cell cycle, we are going to see that there are many, many of these biochemical control which drives the cell cycle. So let's take a look at that, how these mitosis promoting factors actually work in the cell cycle. So the cell cycle control system in a protein kinase based machine. So this is a very interesting idea that what happened, the scientists saw that there is a protein kinase which is known as a cyclin dependent kinase. So the kinases are basically the enzymes which will phosphorylate other enzymes. We have already seen that. And this is a serine threonine kinase. Now, the interesting part is that they found that these series or number of different of these kinases are present in the cell, but they are not in an active form when they are just by themselves. But what happens that if they are dimerized along with another protein, which they named as a cyclin, and we're going to see that there are many, many cyclin. This is a whole group of protein. This is just not one. There are many, many different cyclins, and each cyclin do have a specific, again, I'm repeating, each cyclin do have a specific cyclin-dependent kinase. So random cyclins do not bind to random cyclin-dependent kinases. So you have many, many different cyclins, many different cyclin-dependent kinases, and this pairing is very specific. There are some which can work in a 2 or 3 different pairs, but still they are very specific. So when they dimerize, at that time, this kinase is active and it can phosphorylate, and that is the biochemical control. So what was happening at the mitosis promoting factor? So what the scientists found that this cyclin cyclically is synthesized and degraded, synthesized and degraded, synthesized and degraded. And that is the beauty of the control system. For each of these cell cycle different stages, you have this specific cyclin, which when attached to the cyclin-dependent kinase, this kinase will phosphorylate different proteins, which will make changes into the cell. That's the important part. And here what we are looking at, the mitosis promoting factor means that you have a mitosis-specific cyclin and a cyclin-dependent kinase. When they are in a dimerized form, they will drive mitosis. If this cyclin is not present, that was not there. So if we just go back from this, the previous slide, what happened that in this cell, this mitosis promoting factor means that they have a mitosis specific cyclin, which is along with the cyclin dependent kinase in a dimerized or active form was present in that cell. When this nucleus came into this heterokaryon, all these chromosome condensation is due to the effect or downstream effect of this mitosis specific cyclin and its corresponding cyclin dependent kinase, which they named as a mitosis promoting factor. So that's how this works. So let's take a look detail after this what happened, that you can see in this particular slide that for each phase you have a specific cyclin and its corresponding cyclin dependent kinase. So let's start with right here that we saw the same picture. This is a mitotic dependent cyclin. So it is attached with or dimerized with the cyclin dependent kinase, which is specific for this. And then after the mitosis, this mitotic cyclin is degraded. Once it is degraded, this cyclin dependent kinase is an inactive form. So at this time, this is not going to work. Once it goes into the G1 phase, there are some specific cyclins going to be synthesized, which we call as a G1 cyclin, that will activate the specific corresponding cyclin-dependent kinases that will drive it through the G1. Later, it's going to synthesize this S phase cyclin, which is going to drive it through the S phase. But in the S phase, this G1 or the S cyclin is going to get degraded. And then it will just go, there is no specific G2 phase cyclin because it's not needed. We're going to explain that. And then it's going to go into the mitosis. Now here, what you are seeing, two very important things, just make sure that this diagram is oversimplified. That's the reason you have to be a little bit careful in your concept. So I'm going to go slowly. What you are seeing that the cyclin-dependent kinase is shown in red. And this picture kind of shows you that this is the cyclin-dependent kinase, which is released from the corresponding cyclin, and the G1 and the S cyclin are binding to the same cyclin to which this mitotic cyclin was binding. That is not true. Remember that you have many different cyclins, and this is just a generalized diagram, or these are the generic cyclin-dependent kinases, but G1 cyclin will bind to its specific cognate cyclin-dependent kinase, S-phase cyclin will bind to its cognate cyclin, not the mitotic cyclin. That's how it is depicted right here. So that is a very important concept. So this basically drives the cell cycle and on a regular way that each of these cyclins are degraded. So another important thing, remember that this is also very important, that definitely a note about that. No matter which type of cyclin-dependent kinases are there are many CDKs or cyclin-dependent kinases. Their level do not go up or down during the cell cycle. So all the cyclin-dependent kinases kind of pretty stable all throughout the life of the cell. Yeah, if the protein ages and gets denatured, they are going to get degraded. That's a different thing. But the cyclin-dependent kinase level remain the same. The only thing that is cyclically synthesized and degraded are only the cognate partner, which is the cyclin. So mitotic cyclins are synthesized, doing their function, degraded after that. Then the G1 or S cyclin are also synthesized in the corresponding phase. They're going to get degraded with that. The beauty of the system is that, think about that one protein, which is the CDK, that level of CDK is the same all throughout the cell cycle, but it is active, inactive, active, inactive, just due to the partner which is synthesized in a specific cycle of the cell or phase of the cell cycle and degraded afterwards. So the level of the cyclin go up and down, but the level of the CDKs remain constant throughout the cell cycle. This is a very important concept, and make sure that you will absolutely have questions in your quizzes and exam on this one. That CDK level don't fluctuate. I'm just making sure that everybody is good with that. Everybody good with that? That concept? Good? Okay. All right. Let's move to the next one. So here we are looking at the core of the cell cycle control system from your textbook. So here you have this G1 phase, and what you are looking at, that right here is the rise of the G1 and S phase cyclic dependent kinases activity. Now remember that, very important, do not confuse with the previous slide with the amount present in the cell versus activity. So what I said in the previous slide, that the CDK level remains the same, their activity decrease, increase, decrease, increase throughout the cell cycle. So two are different concepts. Here we are looking at the activity of the CDKs, where the G1 S phase cyclin-dependent kinase activity increases during the G1 phase, and then gradually it will decrease in the S phase. Whereas the S phase cyclin-dependent kinases, their activity will rise during the S phase, and you can see that it covers pretty much the G2 phase, and it goes down into it starts going down in between the G2 and the mitosis and completely gone by the end of the mitosis. So you can easily see that there is no specific G2 or GAP2 specific CDKs, because other CDKs overlap that area. But the level of this mitotic CDKs rise from the late G2, and the amount is highest in the mitosis, and its level will go down at the beginning of G1. Another activity of the protein is shown, which is not a cyclin-dependent kinase. This is a APC. So this APCC activity, this is an ubiquitin ligase and proteasome complex. So what is this? We are going to explain. So as we are seeing that the cyclins and the cyclin-dependent kinases activity is going up and down, the activity of these APCC activity means that this is an ubiquitin ligase, and what it does. This ubiquitin ligase is going to add ubiquitin residues onto the proteins. And if you just remember from your biochemistry that if the ubiquitin moieties are added to the protein, it is destined for degradation. And this degradation happens in a very large protein complex known as proteasome. So I'm again repeating, any protein that is needed to be degraded in the cell, one of the ways is to that adding this ubiquitin moieties onto that, this chemical, and then it's going to get degraded. So what is happening right here, that at the towards the end of the mitosis, that this is the metaphase anaphase transition, these APCC proteasome activity degrade all these previous phase CDKs so that this can move into the next phase. Why this is so? Because this is known as APC, or anaphase promoting complex. See that right here is the metaphase anaphase transition. And only if that anaphase promoting complex is active, the cell can move through this checkpoint from metaphase towards the next one. And we are going to talk about that, how nicely this APCC activity or the ubiquitin ligase degrade a specific protein so that the mitosis can move from the metaphase into anaphase. And we're going to see that activity. So this is basically the overview of the core of the cell cycle control system. One thing, make sure that we understand the principle. And although we are talking about this G1 S phase cyclin and CDKs, S phase mitotic CDKs, each one of them is a number of different individual cyclins. Their names as cyclin A's, cyclin B, cyclin D, E. There are many cyclins in the metazoans which are in your textbook, and specific numbers, CDK 1, 2, 4, 6, like that. And you do not need most of these specific names. If you just remember that what is the principle, that is good enough. All right, so let's move to the next slide. So next clicker question is, which of the following statement is not true? Cell cycle phase-specific cyclins are synthesized and degraded in a repeated fashion. Cyclins are synthesized and degraded in every cell cycle. G1S cyclin amount rises in the G1 phase and will fall in the S phase. And G1S cyclin dependent kinase CDK's amount rises in the G1 phase and will fall in the S phase. Let's take a look. About 15 seconds. Good. Make sure that you can pull that. So I'm stopping it now. All right. All right. So what I see is about 60% answered D, which is the G1 S cyclin dependent kinase amount rises in the G1 phase and will fall in the S phase. Actually, that's the correct answer. And make sure that I specifically put that question because I discussed that just before. And this is all, you are almost going to see this question in your exam. So remember one thing, let me go back and explain this, why this is important, because this is a very important concept. Remember that, as I said, that all these CDKs, or this right here, CDKs, cyclin dependent kinases, no matter what, the level or amount of the CDKs remain constant throughout the cell cycle. The CDKs, or the partners in red right here, they don't go degradation and synthesis. Only their cognate partner, which is the cyclins, they are degraded and synthesized. So CDK's amount remain the constant. And here, again, I specifically mentioned that what you are looking at, that these are all CDKs, and you see that the amount going up and down, it is not the amount, it is the activity that I pointed out. So just make sure that this slide, this is the reason that is very important. Activity means that here you have the G1 S phase CDKs that dimerized with the G1 S phase cyclin. That's the reason that dimerized form, the activity goes up. But the amount, if this is replaced by the amount of the CDKs, it will remain constant throughout the level. So if I just replace this with the amount, it will be the same all throughout the cell cycle. So remember that although they are present, the activity goes up and down. And this is a very important concept that I mentioned, which is different from the previous slide. All right, so let's move on. All right, so here next we are going into a little bit deeper into the cell cycle regulation. Looks like a very, very busy slide, but it is, if you just take your time and just understand that, it is not very, very complicated as such, although it can be, but we will just keep it at the level that it is not terrible. So right here, let's start from here. So here inside, you can see that you have your G1, early, mid, late G1, then S, G2, and the mitotic phase. So at the first, the cell just exited from the mitosis, and here the DNA pre-replication complex assemble at the origins. And what we mean by this. So as you know, that the DNA needed to be replicated from a particular place. This is from your biochemistry. And this is known as the origin of replication. You need a good amount of proteins to be assembled in there. And I'm not going to go or ask anything about this origin of replication, but just saying that what it is needed. You need to separate these two strands of DNA, and there are a number of different enzymes that is needed. There are topoisomerases, helicases, primases, many different types of cofactors, coenzymes needed to do this. So this all start to assemble during this G1 phase. Remember that they are still much, much farther away from the synthetic phase, which is the S phase. And the replication of the DNA will not start until the cell passes the restriction point or the checkpoint of the G1 phase. But the machinery to do the synthesis start assemble at this stage. And right here, we will come back to that. As I said, this is a G1 cyclin CDK inactivate the CDH1, and this is the remnant of the protein that is from the previous part. So we will come back to that once we go through the cell cycle. So in the next part, what we're looking at, the G1 cyclin CDK activates the expression of S-phase cyclin CDK components. So you need a specific cyclin that will drive all the expression of the proteins that is needed in the S phase. And that is synthesized, or the S phase cyclin is synthesized in the G1 phase. And at that time, the G1 cyclin CDK will activate that. So next, what happens that the G1 cyclin CDK will phosphorylate the S phase inhibitor. In your lecture, I just pointed out just to make sure that you understand that with some red arrows, additional annotated that. So this G1 cyclin CDK complex is going to phosphorylate this inhibitor. But before going to the inhibitor, what is happening? Let's explain that step by step. In this particular phase, you have the G1 cyclin CDK complex. This is an active complex, G1 cyclin CDK. That will synthesize the S phase cyclin as well as CDK. So right here, G1 cyclin CDK activates expression of S-phase cyclin CDK components. So again, activates the expression means these genes are activated, transcription done, translation done. You have this S-phase cyclin CDK complex made already in the G1 phase. But there is a difference. Although they are made, this S-phase cyclin CDK will not be active until it passes the restriction point and go into the S phase. They're already synthesized here, but they're inactive. Why they're inactive? Because there is an inhibitor which is also synthesized in this phase, attaches to the S phase cyclin CDK complex. So the reason that this is done, because synthesis of these proteins takes a good amount of time. This is the growth phase, and these proteins are synthesized, but also along with this inhibitor is synthesized, which is being attached to this. Then what happens when the cell asks the questions that if everything is okay in the cell has grown, the cell has enough amount of nutrients, and the DNA damage, there is no DNA damage, it will pass the restriction point. And at that time, what happens that it will activate, when it passes the restriction point, an SCF proteasome complex. So SCF is basically a specific ubiquitin ligase that will attach the ubiquitin to the inhibitor that will be degraded by the proteasome. So now the S-phase cyclin CDK is devoid of the inhibitor, and this S-phase CDK cyclin is now active. And this will activate the pre-replication complex. So what happened to the pre-replication complex, or why it is right here, which is already assembled? Although they were all assembled in the G1 phase, there is some phosphorylation needed for them to be activated. So they are just sitting on the DNA, but in an inactive form. Once this inhibitor is degraded by the SCF, the S-phase cyclin CDK complex is active, and this kinase will be able to activate the different pre-replication complexes by phosphorylating them. Once they are phosphorylated, this will be the replication of the DNA will take place. And then at the end right here, you can see that here a mitotic cyclin CDK will activate the early mitotic event, which will synthesize all the different proteins necessary along with the assembly of these chromosomes. They will congress in the metaphase plate. And next to that, there is also a very important thing, and this is very, very important. There is another ubiquitin ligase, which is the APC-CDC20, which is also an ubiquitin ligase, will attach the ubiquitin to a protein called securin. Once that is degraded, this cell, which is in the metaphase, will go into the anaphase. If you remember from the slides that we talked about this, that what is this APCC? That is your anaphase promoting complex, which can lead the cell to move from the metaphase into anaphase. So let's revisit that. And this is the first thing that the scientist found out, that what is going on in here. So if you look at this slide, particular slide, let's see right here. Right here. So you can easily see that the effect of this APCC activity increases just at the junction of the metaphase-anaphase transition. And this is called the anaphase promoting complex, which will lead to the separation of the sister chromatid by the degradation of a protein, which is securin. So what is securin? Now, let's take a look that this is a slide from the 3.2 part 1. We're not going to look at any of them. Just we are pointing out, if you remember that, this is a cohesion complex, and we talked about that. Think about that. You have two bundles of wood, which are the sister chromatid. They are attached together by this rope or anything. This securin is a protein which is in the cohesion complex. Here you have the securins. They keep the two sister chromatids together. And this is up to the metaphase. From the metaphase, when they need to move into the anaphase, these two sister chromatids needed to be separated. To separate them, the first thing that is needed is to degrade that securin complex in the cohesion complex. So this attachment of the cohesion complex come out, and the sister chromatids can move individually to the each pole. So that is being shown here, that it degrades the securin so that this bundle are free, the two sister chromatids are free, and in the anaphase, it can move. So that is your anaphase promoting complex. And then what happens, that the APCC CDH proteasome also will degrade the mitotic cyclin. So from the G2 phase, you needed the mitotic cyclin to promote all these different activities. They will be degraded. Also, at that time, that you have this proteasome complex that is produced in this phase. So let's see that at this point, what is happening. Remember that when you have this proteasome complex, you do not want that proteasome complex to be hanging around in the next or the G1 phase, because it's going to degrade other proteins, which is not needed. And at that time, this is the step that we omitted. The G1 cyclin CDK will inactivate the CDH, which is this proteasome complex, which basically was responsible for degrading the proteins which were responsible in the anaphase and in the mitosis. So that's your cell cycle regulation. It is much more complicated than that, but for the purpose of your exam DNA and for the scope of the course, I think that if you understand up to this level, that is good enough. Your textbook will is in much more detail. So now what we are going to look at, that specifically, we are going to look at some specific things that is going to happen. And again, we are going to look at a little bit more detail, but not too much of detail, because things can get complicated very easily. So here, the first thing, that how this G1 cyclin CDK will activate this S-phase cyclin CDKs, and how this S-phase cyclin CDKs work, their activity, and then how this S-phase cyclin CDK will promote the pre-replication complex into an active complex. So these are the three steps here, 1, 2, and 3. We're going to take a look in a little bit detail. So we are looking at the first, the G1 cyclin. So here you can see that the cyclin is replaced by a cyclin D and CDK4 and 6. So as I said, that although we are naming the G1 cyclin, S-phase cyclin, or mitotic cyclin, there are many different cyclins, and one of those cyclin is a cyclin D. And this cyclin D can attach to a CDK, which makes a cyclin D and CDK4 complex. This is one complex. Although it is shown as 4 and /6, there are literally two different complexes. One is cyclin D and CDK4, another is cyclin D and CDK6. So the cyclin D can bind to two cognate partners. One is CDK4 and CDK6. That's it. So these proteins also activate another protein. And let me talk about that. What is that other protein? There is a protein in the cell called RB. And the name is derived from retinoblastoma, which is a cancer of the eye, or basically in the cells in the retina. And we're going to talk about that protein. So this RB protein is produced in the cell in an active form. And this RB is bound to another protein in the cell, which is also produced constantly. So again, remember that RB is produced in the cell in the functional form constantly. E2F is produced in the cell in the functional state constantly. Upon production, the RB naturally binds to the E2F. And at this point, the E2F is in an inactive form. Why? This E2F is a transcription factor. When this E2F is bind to the RB, it cannot have its transcriptional activity. So RB plus E2F, it's an inactive complex. So let's see that in the G1 phase, what this cyclin D4 and the cyclin D6 does. It will phosphorylate the RB. Along with that, there is also another cyclin. This is also a G1 cyclin, a cyclin E and CDK2 complex. This will further phosphorylate this RB. And there are multiple phosphorylation sites on the RB. Once the RB is phosphorylated, it's going to change conformation and release that E2F, which was bound right there. So now E2F is free, and this E2F is now active. So the free E2F is always active. This is a very important concept. Remember that. E2F bound to RB inactive, E2F in the free form is active, and this will drive the gene expression because this is a transcription factor. Interestingly, see that what it is doing. E2F will, this is a protein, will bind to upstream of its own E2F gene, and it will make more and more E2F. That means there you have more and more expression. It will also upregulate the cyclin E production. So you are getting more phosphorylation of the RB and more activation of the E2F. So you can see that the E2F itself can feed into the system to auto-activate. So that's the important function of this. And this E2F will lead to a variety of expression, and these expression of these genes, other than this is just the auto-activation, but other than that, it's going to phosphorylate, transcribe, or help in transcription of many of these genes. And those are the genes which will express many of the proteins needed in the S phase. So that's how the G1 cyclins activate. Now, let's take a look at what is going on a little bit more detail. So here you have a piece of DNA. This could be a cyclin E or cyclin A. Now, cyclin E is the same one that we have seen right here, cyclin E. This is the auto-activation cyclin. Cyclin A is not needed in the next phase. And at that time, what is happening, that RB and the E2F is bound together. RB is inhibiting the function of E2F. Now you have this cyclin D and the CDK4, cyclin D CDK4 and cyclin D CDK6 complexes right here. They will be active with the growth factors. Now think about this. What we talked about that in this G1 phase, the cell senses that whether they are active or not, in the sense that it's checking the environment. Is the cell big enough or is the environment good enough? That means they need growth factors in enough amount so the cell can grow, and these growth factors will activate the cyclin D CDK synthesis and expression, and this will phosphorylate the RB. RB separates out from the E2F, and E2F will drive the expression of cyclin E more and cyclin A, which is the next phase cyclin. So you can see that cyclin E and the CDEA, cyclin E CDK2 or cyclin A CDK2, that will promote the transition from the G1 phase to the S phase. Also, it will lead to the centrosome duplication. So the mitotic spindle apparatus, you have this mTOC, and the organizing center is also going to duplicate that, and the spindle pole will move towards the two opposite poles of the cell that is necessary for the next phase. So it starts pretty early, so you can easily see that this is the way that the G1 cyclins work. Any questions so far? All right. So, okay, great. So the next one is just an idea that what happens. You were seeing that this is a dilated pupil, and you can see that this is the tumor developmental of the eye due to the mutant of the RB protein. And historically, it is named because the non-functional RB was found in this eye tumor, which is the retinoblastoma. So that's where they were discovered, and that's why, although this is present in every cell, but it is named as retinoblastoma because it's implicated in that cancer, and it was discovered there as well. So what happens in this case? If you have a mutated RB or a non-functional RB, it cannot bind to the E2F. So as it cannot bind to the E2F, remember that E2F is a transcription factor. It will be inactive only if it is bound to the RB. So E2F is always active. And what it means, if it is always active, means it's always going to drive the cell cycle. And that's the main problem. So if you can easily see that here, the cyclin D CDK growth factors, they only upon phosphorylation, the RB separates out. But if RB even cannot bind to the E2F, the E2F will be in this form, and it will immediately after the cell comes out of the mitosis, before it can check whether there is enough growth factors, whether the environment is okay, whether there is no DNA damage. Before it can sense that, it's going to produce all these next phase cyclin, and it will go into the S phase without checking the environment, and uncontrolled cell replication leads to cancer. So that's the reason that a tumor development eye due to the mutant RB protein, which cannot bind to the E2F, and E2F is always active. So that's your part one. So in the next part, we are going to see that what the S phase cyclin inhibitor is doing. So if you just look at that, we are just pinpointing out to this complex, that how this is activated, whereas the S phase cyclin CDK inhibitor attached to it. So right here, what you are seeing, that here you have the S phase cyclin attached to the CDK, and there is a protein, now you know the specific name. This is known as the SIC1. So the SIC1, when attached to the S phase cyclin, this S phase cyclin CDK complex, although dimerized, it is in an inactive form. So the G1 cyclin and the CDK complex, active complex, phosphorylate the SIC1. Very straightforward process. Phosphorylate this SIC1. Upon phosphorylation, what can happen? It can be degraded by polyubiquitination. So this is an important concept. Direct phosphorylation doesn't remove it from the CDK. Remember that the previous one, we remember right here, once the RB is phosphorylated, it releases the E2F, and that's the process. Here, the SYK1, the inhibitor, is phosphorylated, and the phosphorylation doesn't release the S-phase cyclin or the CDK, but the phosphorylated form can be added with the ubiquitin moieties. So that leads to the polyubiquinolylation, and that will lead to the degradation of the SYK1 by the proteasome. And once this is degraded, the S-phase CDK cyclin is active. That will lead to the DNA replication complex, the promoting complexes, which are the replication complexes at the origin, to be active, and it will drive the DNA replication. So very important. Phosphorylated SYK1 can be ubiquitinated, and that will be degraded by the proteasome only. Unphosphorylated form cannot be degraded. So that's the activity, that how the inhibitor degradation can lead to the S-phase cyclin CDK complex activation. I just repeated it many times for the concept. So the third phase is that it is much more complex, and you do not need to remember or memorize any of the specific names of these proteins. If you just remember the overview of this process, that how the S-phase cyclins do work, that is good enough. So as I said, that right here, you have this origin of replication, and there is a protein complex which binds to this origin of replication, and this is an inhibited protein complex. That means, say, that in the G2 phase or the M phase, the DNA cannot replicate. The DNA cannot replicate because of this complex, which is an inhibitor and doesn't allow to replicate the DNA, because you do not want the DNA to be replicated in the G2 phase or during the M phase. So what happens that in the G1 phase, there are several different other proteins which are loaded onto this complex. And you can see that if you remember from that, again, it is not required. I'm just saying that helicase is one of the enzyme which actually unwinds the DNA. So that is important. So the double-stranded DNA is unwind with that. So these are loaded onto the DNA, but still this is an inactive form. So let's see that what happens in the S phase. Remember that in the previous slide, we saw that the S phase cyclin is activated by the G1 phase cyclin when it enters into the S phase. Now in the S phase, this S phase CDK is going to phosphorylate these complexes, and these are the inhibitors. So once these inhibitors go out, you can easily see it will also have other kinases which will phosphorylate some of these inactive proteins on the complex. See that right here, if you look at that, this MCM helicase is binding onto near the origin, but once they have this phosphorylated form, they are the active form. So it does two things. Phosphorylation of the inhibitors release the origin of replication, and it allows the replication complexes to promote DNA replication, and it also, this S-phase CDK cyclins will activate other kinases like these DDKs and other proteins, which will allow the DNA to be replicated. And here you're seeing the complex, or the origin of replication complex, which to end of the fork, which will allow the DNA replication. So this is your function of the S-phase cyclin. You do not need individual proteins, just the overview of that, how the phosphorylation of the inhibitor makes them move out of the way from the origin of replication. That's number one. And also some of the component of the replication being phosphorylated makes them active for replication. That's it. Nothing else is needed. So the next part that what we are going to talk about, we already talked about the checkpoints, and let's take a look at these checkpoints. All these checkpoints ask questions, and they will halt the movement into the next phase if it is needed. So let's first take a look at this, that you have an unreplicated DNA checkpoint, a spindle assembly checkpoint, and a chromosome segregation checkpoint. Other than these three, you have 4 DNA damage checkpoints. And let's see that one at a time, that what they are doing. So the first thing is an unreplicated DNA checkpoints. So right here, this happens before the cell enters into the mitotic phase at the very late G2 phase checkpoint. So what it means that before the cell enters into the mitosis, The unreplicated DNA checkpoints wants to make sure that before the cell divides, it has all the replicated DNA. If it doesn't, it will not allow the cell to enter the mitosis because it will be detrimental to the cell because you're losing genes because it's not replicated. So how this is determined is by a protein called ATR. So this ATR looks for, and again, that is not needed. The mechanism is not needed at all, but ATR look for single-stranded portions of the DNA, which can lead to the idea that there is some unreplicated DNA is in there. So it just slides along with the chromosome for looking for unreplicated region of the DNA. Once it is activated, it induces the expression of another protein called CHEK1. And this CHEK1 inhibits a particular protein. Now you know that this is CDC25C. So it's right here is CHEK. So make sure that you understand that what it means. ATR induces the expression of CHEK1. CHEK1 stops the function of CDC25C, which is responsible for activation of the cyclin A, CDK1, and cyclin B, CDK1. So this cyclin A, B, and along with the CDKs ones, these complexes are responsible for moving from the G2 to the M. So again, remember that it means that these are your M phase cyclins. These are specific name, but these are M phase cyclin. So CDC25 will block the function of these M phase cyclin, so the cell cycle will not move into the M phase. And this is done by the protein ATR if it senses any unreplicated DNA. So it will arrest the cell cycle at the M phase entry. This is one checkpoint. The next checkpoint is the spindle assembly checkpoint. And this is done by the MAD2 protein complex. And this works on this APCC CDC20 complex. And see that how beautifully it's orchestrated. You now know that this is the anaphase promoting complex, and this APCC, and this was the C/C. Now you know it is CDC20. This complex ubiquitinylate the securin. That means it will separate the two sister chromatids so that they can be separated into the anaphase. But if the chromosomes do not assemble on the spindle at the middle portion of the cell or the metaphase plate, this MAD2 will not allow the movement of, or the event will not move from the metaphase to the anaphase. So that is your checkpoint. So a polybutylation of securin is going to be stopped by MAD2. Remember that the securin is needed to be degraded so the two sister chromatids separate, and that is halted by MAD2. The next portion, what we are seeing, is the chromosome segregation checkpoint, and this is done by this CDC14, and this is done by this APCC polyubiquitinylation of a B-type cyclin. So after the mitosis is done, it is needed that you ubiquitinylate the anaphase promoting complex at this point, and that will not be done, or the cell will not move into this telophase or divide the cell until the chromosome segregate in two poles. And this also will help with another protein, which is called a SIC1. So these are the checkpoints that it will not move into telophase from anaphase if the chromosome do not segregate towards the two poles. So these are the three main checkpoints. Along with that are event checkpoints. So the 1, 2, and 3 event checkpoints. Other than these 3 event checkpoints, you have 4 DNA damage checkpoints, and all of them are similar. So let's take a look at that. So there is a protein called ATMR, and you can see that this ATMR is present in everywhere. This ATMR looks at DNA breaks, single double-stranded breaks. So it scans the chromosome, and DNA breaks means that it is damaged DNA. So once it binds to the damaged DNA, it will activate a protein called P53 or stabilize a protein called P53 everywhere. And this P53 will induce or activate a P21 CYP, this complex. So the function of the P21 CYP is to inhibit the cyclin D CDK4 or cyclin cyclin D CDK6 complex in the G1. So if they inhibit the G1, these are the G1 cyclin CDK complex, it will be arrested at this point. If the same way the DNA damage is occurred, if the cyclin E or A with CDK is inhibited, it will stop the entry into the S phase. Right here, if the cyclin A CDK2 complex is inhibited, it will stop the movement from S phase to the G2 phase. And right here, it will not allow the M phase entry. So you have 1, 2, 3, 4 DNA damage checkpoint. Now, interestingly, this is the same way that ATMR, P53, P21, which in every step are inhibiting the cyclins. But along with inhibiting the cyclin CDK complexes, there is another additional checkpoints which are with this bluish color, this cyan color. So what you are seeing, ATMR will also induce the activity of a check 1 and check 2. You have already seen check 1. Here is another additional protein. Check 1 and check 2. Here you have a check 1 and 2. They will induce another protein, CDC25A. CDC25A. And this protein also inactivate another protein, which is CDC25A. So you can see that check 1 and 2 inactivate CDC25. Check 1 and 2 inhibit the CDC25. So what is CDC25? Remember that CDC25 is also an activator of the CDK cyclin A. CDC25 is an activator of cyclin E and A and the CDK complex. So check 1 and 2 will Are you guys good now? At least?
Yeah.
Yep.
Good. Okay. All right. So that's it. So what we were trying to say, that just reinforcing the same thing, that the check 1 and 2, that what you are seeing, that these are the additional 2 things, because this is very important. You don't want to replicate damaged DNA, and you do not want any DNA to enter into the M phase if during replication there is any DNA damage. So that's the reason that you have these 2 additional checkpoints. So, and the quick thing, the last thing that is important is that how this works. Very, very simple, that P53 is a transcription factor, and it's a 53 kilodalton protein. That's why it's P53. And that P53 is very unstable. So that means that it is synthesized in the cell and it's automatically degraded in the proteasome time to time. On the other hand, what happens that if there is any DNA damage sensed by the ATMR, at that time, it stabilizes the P53 by binding to it. And that will activate this P21 synthesis, induced synthesis. And this P21 CYP binds to the cyclin CDK complex again as an inhibitor, which stops the cell cycle or arrests it at that particular checkpoint. So what happens that P53 is a very, very important transcription factor, which if it is not properly regulated, then it can lead to many different cancer, many different cancer. So that's the reason that P53 is called the guardian of the genome, because all the DNA damage is detected by P53. Not detecting it can lead to uncontrollable cell growth or the cell cycle. So that's the last question, and I will let you go, is which of the following statement is not true in a child diagnosed with retinoblastoma? There is a mutation in the RB gene. The child can't make a functional RB protein. The E2F transcription factor is inactive due to non-functional RB. Cyclin E CDK2 is functional in the child. All right, stopping the poll now. All right, make sure that you press it. All right. So, yep. So this is the correct answer. 63%. Make sure that the E2F. So here there is a mutation in the RB gene. That is correct. The child cannot make functional RB protein. That is correct. That's the reason that it cannot bind to E2F. And the E2F transcription factor is inactive due to non-functional RB. That is not true. And we'll come to that. Cyclin E CDK is functional in the child. That is correct. It has nothing to do with that. But why the E2F factor inactive due to non-functional RB is not true? Remember that this is a very important slide right here. If RB, which is non-functional, cannot bind to the E2F, E2F is always active. That means that it is constantly going to transcribe the genes and the cell cycle will go on and on. That's uncontrollable cell growth, and that will lead to cancer. So that's basically what it is. So good, everyone, so far? Anyone want to say anything? Anything specific? No, any questions? Just at least one or two person can tell me that so that I can just definitely hear at least. So all good? I'm just posting it in the chat as well. If it is all good, I will let you guys go. Okay.
I actually have a question real quick.
Yeah, sure, absolutely. Mm-hmm.
Okay, so on the very last slide, the CIP binding, does that prevent SIC1 binding so the inhibitor can't bind to the CDK cycling complex?
So right here, but
yes.
Oh, the cycling CDK right here. Oh no, this is
the CIP. So is the CIP preventing SIC1 binding? No.
This is an entirely different site. This is an entirely different site. Yeah, it looks like, well, I completely understand. So it looks like that the S-phase CDK cycling right here, it is binding to this inhibitor as well. No, this is an entirely different, so everything remains the same, but the inhibitors, which is right here, is the P21 CYP. This binds to a different area. So everything is synthesized in a proper way, and it will ubiquitinolate the same way as such. But this will bind, it will although look like the same way, but no, it is just binds to a separate, in a different area. Yeah.
Okay. Yeah. Thank you.
Yeah, sure. Anyone else? Any questions?